Thank you. I'm talking about the tumbleweed. So the problem is movement. Movement is inextricably linked with life and at all length scales. So at the molecular level, movement is created by protein motors, very sophisticated machines that operate with amazing properties. So how do we understand these machines? Well, the normal approach is to take a top-down approach, get structures, get mutants, measure enzyme kinetics, make beautiful movies. But when you look at these movies carefully, there's always a miracle step, energy transduction. We don't actually understand how these motors transduce energy. Historical interlude. A long time ago in the 90s, Heine Linker and I were both in the School of Physics at UNSW. We had coffee one day. Heine was working on quantum ratchets. I was working on motor proteins. And we talked about what we did, and we sort of discovered the principles for these two systems might actually be the same. Okay, we became friends. Heine took a job in Eugene, Oregon. We used to call each other on a Saturday morning and talk about things. He's still working on quantum transport, I'm still working on proteins. Wouldn't it be nice to do something together? What could we do that would be so spectacular, get us into nature or science? And the idea came up, build a motor protein from scratch. So, first we had to get ideas, how do we do this? And secondly, we need money to do such a project. One day I was waiting for a train to go into the city Friday afternoon. Train didn't come on Summer Hill Station, and the idea of the tumbleweed came up. So some kind of a circular protein with DNA binding protein modules that are ligand-gated. And this was the tumbleweed. Took about 2 days to realize there aren't any such monomeric DNA binding proteins that are ligand controlled. So we had to rethink, and that design there is the tumbleweed. Okay. Who's crazy enough to fund such a project? You guessed it. So that's our team. We got the money. I'm told we were at the edge of what was fundable. So Heine's lab, he moved to Lund, my lab. Nancy Ford in Vancouver, single molecule person, and Dec Wolfson, a peptide designer, protein designer. Okay, so our aim is to understand how protein motors work by taking a synthetic approach, build it from the bottom up and see if we can figure it out. So what do we need? We need to have some kind of a philosophy. Ours was use a modular approach. This is before serious protein ab initio design. Steal things where we can, functional components from biology, and assemble them, integrate them, and if we think we understand how it works, it should work. Okay, and we set our own rule that we didn't want to use existing motor parts because that would be considered cheating. Okay, so what are the principles for a molecular motor? We want something that has a source of energy, it's far from equilibrium, it's got to have thermal fluctuations. Diffusion is key to making these things work. We need unidirectionality, we need processivity. If it falls off the track at a molecular level, it's lost forever. And timing. Separation of time scales, separation of energy scales, these things are critical. The tumbleweed. So tumbleweed is not autonomous, it's a clocked walker. We tell it when to take a step, it does it. So, first chicken thing was we chose DNA as a track. It'd be better if it was a protein, but DNA is easy to buy, cheap, periodic, easy to manipulate, and there's lots of DNA binding proteins. So what is the tumbleweed concept? The idea was having 3 legs. This is to get asymmetry. Each foot binds to a specific site in the presence of a ligand. We have 3 such sites, so we needed to have a 3-legged protein. And we need a microfluidic system to deliver the ligands. The ligands are going to supply the energy, the timing, everything. Okay, what's happening here? Good. So, tumbleweed binds in presence of ligand B to site B. Then we add ligand C. By pure diffusion, it will find site C and bind to it. We've designed it so the legs are too short for it to actually step backwards. We remove ligand B, we add ligand A. It can reach the forward site, but not the rear site. So this is the idea. Okay, so it's a clocked walker, it's a ratchet. There's nothing powering the movement. The movement is diffusion. We're just rectifying it. It's asymmetric and precessive. Can we make it? Three-legged proteins are not so common. So Roberta Davies is the star. She really made this work. So the feet. We just chose typical bacterial repressor proteins. So they each have different ligands. This one's a metal, this one's tryptophan, this one's SAM, and they bind to DNA specifically. The legs, we used coiled coils. Dick Wolfson was in our team. This was the obvious choice. We ended up stealing from nature rather than a synthetic one. Natural ones behaved better. Sticking it all together was quite complicated. We ended up using SpyTag SpyCatcher. We had earlier successes, but this really works well, and you end up with a covalent structure. So we express one component, Tumbleweed 1. It forms a dimer naturally, and it's got the SpyCatcher. We express the second component, which has two legs and SpyTag in the middle. We mix them together, and miracle happens, we get a covalent structure. Okay, do we know we have this? So the dimensions are roughly 10-ish nanometers per leg. Knowing that we actually had it, we had to get evidence. So the work of Roberta Davies, Neil Robertson, and Chu-Wai Liu. So, STS page, we purify the individual components, we mix them in excess of Tumbleweed 1, we get the complex, we purify it, we have the complex. So that works. Mass photometry, we get the sizes of the components, it's the right size. So everything is dimeric, but it's a single copy. And it's a flexible structure. Crystallizing it is not possible. We've tried cryo-EM, we can't see it, but Sexax works well, we get a nice pattern, and it's an ensemble, but it's dominated by two forms, a bent form and a straight form, and the residuals are very low. So we've got it. Does it bind to the track? This is the work of Neil Robertson, Roberta Davies, and Aaron Lyons in Nancy Ford's lab. Okay, so what happens? We have surface plasmon resonance. We have thousands of these, but it binds, each leg binds to its track in the presence of the correct ligands. There's a little non-specific binding, but it's not so bad. It's fine. Affinity constants, dissociation constants in the nanomolar range, which is what we want for a single molecule walker. And if we use mass photometry, we can see that if we add the DNA, it shifts. And if we add a two-site track, it shifts even more. And we get no cross-linking of tracks. It just binds to the same track. All good. Nancy Ford's lab made a 8-copy track with the 3 sites, stuck it in a plasmid, linearized it, and Aaron Lyons did AFM, and there's 8 bright spots in the presence of tryptophan. That's the tumbleweed sitting on the DNA of the linearized plasmid. It's exactly in the correct location, so it binds DNA. Optimization, this took a long time. So this is Chu-Wai Liu and Neil Robertson. Too many SPRs, too many kinetic measurements, getting all the timings, all the affinities, the off rates, the on rates. A lot of work, we optimized everything. Okay, next question. Does it actually walk? Can we actually see this? It's small. So this is the work of Patrick Nielsen and Niels Gustafsson in Heine Linker's lab. So we're changing ligands, and we're changing them in pairs. This is what we expect to see. The steps are about 15 nanometers, so it's small. It's going to be hard. It's on a single strand of DNA, so it's flexible. Okay, the first thing is the microfluidic device. So we've got to be able to change ligands arbitrarily. We have 3 sets, AB, BC, CA. And this is just fluorescence to show that we can switch it very fast. This is the observation chamber. We're using turf. So the first set of experiments to show that it's actually moving are on a short track, 4 sites: trip, DTX, met, trip. They're the 3 legs. We put a fluorophore at the end, a fluorophore here, it's attached to a cover slip, and a fluorophore on the tumbleweed. So what's going to happen is, it's on the tryptophan site. So if this fluorophore is down here, you get fret. If the fluorophore is up here, we get fret. If it's in the middle, it just fluoresces. So we're only going to excite the tumbleweed and look at fluorescent signals. So in Heine's lab, we have an optosplit system which allows us to see 2 things at once. So we can't do everything. What we do is experiment 1, where we see fret if it's at the bottom, fret if it's at the top, and fluorescence from the trip of tumbleweed if it's in the middle. The second experiment is we set it up so we see fret on the bottom as cyan, fret at the top in magenta, and in the middle we see nothing, just darkness. So you're going to see chymographs for these 2 based on turf, okay? So what will emerge here is chymographs and chymographs. There's about 250 tumbleweeds, single molecules, walking on a single field of view. And over here there's about 200, and this is a composite of about 2 fields of view. So we select the tumbleweeds by looking for spots that are co-localized, tumbleweed fluorescence, DNA fluorescence. Once we have them, then we track the fluorescence as we change the ligand conditions. So we change the ligands to make it step up to the top of the track. So we should see red here, and we should see magenta here. We then change the ligands again. We're in the middle. We should see green, and here we should see nothing. We change it again. We're at the bottom. We should see red again, and here we should see cyan. We change again. Now we should see black over here, green over here. Change again. This guy should be red. This guy should be magenta, etc. So we just walk up and down. So these are ordered in terms of when we actually lose the signal. Either the tumbleweed has fallen off or it's photo-bleached. We've set up our photon budget so that photo bleaching and falling off happen roughly at the same rate. So if I look across this, we're seeing up to about 12 steps before it falls off or photo bleaches. And on average, they're taking something like 5 to 6 steps. They're not perfect. This is not perfect magenta, black, cyan, black, magenta, but it's doing some misstepping. But overall, it's looking pretty good. So this is what we have at the moment. The cycle we're using here is about 7 seconds, and it's just stepping through. Okay, so it walks. We've achieved our first goal. We have a ratchet that walks, that we can control, and it's behaving itself. How good is it? So the 2 factors we've looked at, this is an unloaded system. We don't have a load. It's not doing anything in terms of work. It's just walking. We look at processivity and speed. So processivity is number of steps times step size, distance in nanometers, and speed nanometers per second. So these are artificial DNA walkers. They're in this region of space, and these are real protein motors, our friends myosin, kinesin, and dynin. So we land here, and this surprised us when we first saw this. We didn't expect to be that good. It's not bad. It's not as good as a real thing. It's beating a lot of the DNA motors. Okay? So the 2 parameters we're measuring, we're not doing badly. Can we improve it? So our initial cycle time was 7 seconds. That was being chicken. Nowadays, we can actually get that to just below 1 second. So we have some data for 2 seconds. And if you're cycling faster and it's still falling off roughly at the same rate, we'll take a lot more steps. We still have some optimization to do. The kinetics can be better. We're playing now with simple things like switching our metal to control one of the repressors, and that speeds up release of the feet, allowing us to go faster. So when we do this, we're expecting to end up somewhere around here. At the moment, we're probably around here, somewhere in between, but that's about the limit we can expect. We're still not as good as real motors, but we're not doing badly, given that we've really started from absolute scratch. Conclusions. Okay. So we've successfully built a modular motor that can actually walk along a track. We can successfully take something like 6 to 12 steps. We can actually do more now on a DNA track. And the energy for our tumbleweed is being supplied by those ligands. It's basically binding a ligand from high ligand concentration, releasing it at low ligand concentration. It's just chemical potential. It's a ratchet. We have no power stroke. And the performance, when not loaded, is approaching that of real protein motors. So we're really excited about having gotten this far. Where are we going? Because of the Human Frontiers grant that really kicked this project off, because we like each other and have kept working together, we've now managed to get an ERC Synergy grant, so the project is supercharged. We're now using ab initio design tools, again combined with modular assembly, and our aim is to build a bipedal, symmetric, mistake on my part, autonomous protein motor. This is going to be much harder than tumbleweed. What are the challenges? The two big challenges are we need to now design enzymes, and that is really at the front of ab initio protein design. And we have to master allostery. Allostery is hard, and we really do not understand allostery very well. Okay, so this is the Human Frontiers team. This is just before the Montreal meeting. Nancy, Roberta over here, Heiner in the back, me somewhere in there, and Deck Wolfson. Who did the work? I've talked about them as they come. Neil, Roberta, Chu in my lab, Aaron in Nancy's lab, and Patrick and Niels. And funding, Human Frontiers, absolutely fantastic to let us even start such a crazy project, and now ERC have picked it up. Thank you.